So genes are kind of like positions for basketball. Center, power forward, small forward, shooting guard, and someone like Magic Johnson, who's the, what is that called? Come on. Yeah, exactly. So, so here we go. These are like in terms of the lactase gene, it's on in childhood, and usually then it's turned off. But in humans, in certain populations, it's left on all the time because of dairying, and human adults could actually drink milk, and it was a good food source. Now, in when we're talking about, and everybody know what's forensics? You know what forensics is? Give a guess. Okay, and what is it used for? Somewhat, right, exactly. So forensics is part of, they call it forensic science. You've all seen, the crime scene stuff. There's a whole bunch of stuff on TV about crime scenes and processing crime scenes. Part of processing crime scenes is if you get DNA, you can usually match it to a suspect and exclude a bunch of others. Just so you know, forensics is not a science. It's actually, it's riddled with errors, I tell you. They put people away for so much stuff that they were obviously innocent. But anyway, DNA is pretty good. So these are the different chromosomes. They're not homologous here, they're just single. There's 22 pairs, and then there's X and Y, which is for sex chromosomes. And there's these things called short tandem repeats. There's stretches of DNA where the bases are paired are repeated over and over and over again, and they're highly variable. That means each person usually shows differences in their short tandem repeats to other people. So these areas are usually, they're called non-coding regions, and that they don't code for proteins. They actually do have gene functions, though, and usually that has to do with gene regulation and how fast genes are expressed and to what degree. So they're repetitive DNA sequences. Now let's take a look alike. So in this case, here's a short tandem repeat for forensics analysis. The repeat is TAGC, TAGC, TAGC. There might be thousands of them in these short tandem repeats. And people usually have a different number of repeats. So someone might have, let's say, 14 repeats, and someone might have 18. And they get this from either their mother or their father. So this is kind of like a DNA fingerprint. This is what's called a gel. And these different bands are different length repeats that have been cut with an enzyme. And we get 2 copies, one from mom and one from dad. And well, let's take a look at this. So here we go. You can actually see it. This one up here, 15 repeats. This is only 10. Move further. Oh, that's it. Okay. So we'll go back to the other one.